And here we go with part 3 of what antibodies can do. This is going to be about how antibodies can be used to fight cancer. For the biochemists in the class, this is going to connect directly to the work of James A. Wells that we followed through. It's also going to connect to what we're going to be talking about later in the class. We're going to be talking about T-cell research, anti-COVID research, but also we're going to be talking about why that takes place at a cancer research center. These are all connected. Remember, the immune system has a role to play in fighting off cancer. And what is the antibody? So, if you have an antibody that binds a cancer cell, you have a good thing. You basically have arrows you can shoot at the cancer. The question is, can they really hit the tumor as effectively as you need it to? But they can at least hit the tumor and you can do different things with them. This whole area is called immunotherapy, and there's a video like you can watch if you want to see a well-produced video of one of these. If I remember, I'll link it to this playlist so that you can watch that as well. But the general field of cancer immunotherapy is a big deal. I've told you about the book Emperor of All Maladies, which I really like. It includes a whole lot of information about using antibodies against cancer. In 2014, this took a big step forward in that Science magazine actually named it the breakthrough of the year, because we can manipulate the immune system. We've learned enough to be able to really do things differently with it than we were able to do before. But of course, it's very expensive. These immune proteins are very complex. Manipulating immune cells or making immune proteins in bulk or delivering them even is tricky, complex, and costs a lot of money. So one of the questions we have to ask is, what is worth going after with this? It's one of the reasons why we don't have that many immunotherapies for coronaviruses lined up, because, honestly, we've been working on it, but it hasn't been the highest priority until the pandemic hit. I look forward to seeing what we'll come up with in the next couple months and years. I'm just hoping it's sooner rather than later. Because antibodies are so adaptable and so powerful, I hold out a lot of hope for what they can do. And I'm going to show you about how antibodies have been used to go against cancer targets, which is, in some senses, the hardest kind of target to go after. It's your own cell. It's just replicating differently. Why should that make any kind of difference on the surface where the immune system can detect? Because the immune system can't go inside other cells. Okay, there's like a Fourth Amendment for cells because you can't go in them. But you do have windows into it, and we're going to talk about a lot of those windows. For right now, we're going to talk about the idea of using antibodies, which have to be outside the cells, to fight cancer from the cell surface. So James A. Wells is part of the team leading the Antibot, and the Antibot is a robot that makes antibodies with phage display, and that should be very familiar to those who have read Delano and Wells. This is like that TV show where you have all the different strands and they all come together in the end. Actually, Tom Clancy novels did the same thing. It's all coming together, okay? The Antibot, you can watch this video, and I'll probably link to this one as well, to see a quick thing of it in action. As you watch it, biochemists, notice that there's lots of things that you've seen. It's doing the same things that you do in lab. It's just doing them in a robotic way. So when you know about how to do these things in lab, you actually know a lot about how the Antibot runs. Phage display really is pipetting things in different places, incubating at different temperatures, shaking things while they wait. It's a lot like PCR is. It's a complex biochemical procedure. The actual lab work is very transferable, and it's what you've already done in the lab. Here's a website for the Recombinant Antibody Network. The antibody is going to be made by this robot, and it basically does phage display on antibody domains. So this is the paper that I showed that I said would tie in. And it's kind of a complex paper. What I want to do is I want to run through this paper and explain it to you. In past years, I've asked people to read it. This year, I'm going to just describe it to you. But this is the kind of process that I want you to be doing, especially for your last couple presentations, especially presentations 3 and 4, will probably involve complex papers with complex concepts. So I want you to see James A. Wells is the last author, the one with the most responsibility on this paper. And this talks about the way that he used the Antibot to go after cancer. What they did is they used the Antibot to generate a toolkit of recombinant antibodies to 7 proteins that are induced by RAS. RAS takes you back to chapter 12 of biochemistry. That's the growth pathways, and remember that RAS is a central hub protein for growing the cells. If RAS gets certain mutations, it's like a light switch that stays on, and it keeps the cell factory running, it keeps the cell dividing. By the way, that sounds a little bit like viruses. You would not be wrong if you said that. But for this case, it's about cancer. If RAS gets a mutation and stays on, the cell keeps dividing. And that's the thing is, what differences does that make on the surface of the cell where the antibodies can get to? His whole job is to systematically look at cancer cells versus non-cancer cells and look for differences in what is on the surface of that cancer cell that we can go after with an antibody. Or vice versa, you can have a difference of something that is on the normal cell but not on the cancer cell. You could use that as well. Look at the end of the abstract. Taken together, this work presents a technological platform for attacking RAS deep inside the heart of a cell from outside the cell. The difference between outside and inside is at the heart of what this is going on. Antibodies are outside reagents. They're being used to attack cancer, which is an inside the cell phenomenon primarily. But there are little differences outside the cell, so you got to find them. This paper actually set up the paper that we're most talking about. This was the 2016 paper, and what it did is they made antibodies, and they were targeting surface differences, and they wanted to look at all the proteins on the surface to try to find the best ones. And so they had to have a model of 2 cells, very similar, but one of them had cancer and one of them did not. The simplest way to give a cell cancer is to put this glycine to valine mutation in the RAS. And so that's what they did. They did RAS G12V. It transforms it into a tumor. And you can see on the left, you can see the morphology of the cell is definitely different. The cancer cell has a different shape. Does it also have different proteins to go with the different shape? So basically, this previous paper, this 2016 paper, goes through and they do proteomics, which means they look at all the proteins on the surface of each kind of cell. They compare the two, and they try to find differences. They found that there was a pretty big difference between the two. So then, in this paper, they narrow it down to particular proteins, and they build antibodies against those proteins to have an antibody that would bind a cancer cell, but not a non-cancer cell. So antibodies that bind receptors on tumors are big business. The basis of Genentech is the Herceptin antibody. I mentioned that last time. It's a cancer treatment, and basically if you have a normal breast cancer cell only has a few of the receptors on it. A breast cancer cell has a lot of those receptors. Now see that that's not tight logic, okay? It's not necessarily necessary. The more necessary it is to the cancer, the better of a target it is. But you see how we're just going with a correlation. More receptors tends to correlate with cancer. But this is good enough to form a therapy out of, especially if you have Herceptin. And by the way, this Herceptin is a growth signal receptor. So there is a good reason to go after it. It's one of the reasons why Herceptin works so well. Herceptin will actually stop the receptors from signaling growth. So not only does it block something, it stops them from growing and actually stops the cancer. So remember that Wells was for Genentech. I believe I mentioned that if you look at the list of where people work, there's always good information there. And what you see is that Genentech is the funder of this research. It was founded on the success of Herceptin. And so they have a good history with antibodies, healing diseases, and making them money. And so they're looking for more like that. So with a complex paper like this, it's usually good to start at the end. So I'm going to skip to the very end of it, and I'm going to have the final section, and I'm going to highlight, if I look at this section, they're talking about CDCP1. It is the protein they found as a cancer therapy target. They looked at a lot of proteins, and they're just talking about one. That's something we can talk about in the future. But when you're trying to understand a complex paper like that, remember, reading out of order is a good idea. Spoilers are not applicable. You want the spoiler that CDCP1 is the ultimate result of this. If you look up and down, you can say that they found that it was involved in a pancreatic cancer. They found that it was involved, they found this protein, and then they looked in cancer protein databases, and they found several examples where there are expression levels on different levels of cancer. And they are saying that the cells were expressing 2.9 million copies per cell. That's a lot. And so that sounds like it's a good target. How did they end up choosing CDCP1? And how did they make a specific antibody, monoclonal antibody, to that specific protein? The cancer therapy, they showed the cancer therapy goal. This is another article. This is a figure from the end of the article. But it's systematically showing you that they are using recombinant antibodies to target and detect RAS signaling. So they're detecting that RAS signaling is constitutively on by detecting the CDCP receptor. This shows you two ways they can use an antibody against it. One way is that they can use for cell killing. Now, for practical reasons, the cell, usually you make a bunch of the primary antibody, but it's hard to label the primary antibody, and it's expensive to make. But because all the primary antibodies have the same constant region, it's actually cheaper to make a whole bunch of a constant region binding antibody that's called the secondary antibody. It's shown here in pink. And that antibody you can make a bunch of, and you can put like toxic payloads on it, and you can attach anything you want to it. So it's just you use the fact that all antibodies have the same constant region to do a primary antibody that binds the receptor and a secondary antibody with the toxic payload that binds the primary antibody. It seems kind of weird, but it's what works. Okay, so if you do this with a secondary antibody that has some kind of toxic thing, then you have that secondary antibody. It's coursing through the body, of course, but it's concentrated at the tumor because it's binding the antibody that's binding the tumor. That's called antibody-drug conjugate cell killing. We actually had, and by the way, these go with our ACS posters. Some of them, you might recognize some overlaps with it. It all ties together. Another thing you can do is you can actually take the antibody, and it's shown here in sort of blue, and you can attach a T-cell activating domain to it. This domain binds a T-cell receptor, and it alerts the T-cell to the fact that there's a tumor cell here. This is called antibody-mediated T-cell activation, and this is kind of cool because you're chaining two parts of the immune system together. You're using the antibody to activate the T-cells. That's kind of neat. And this actually can work pretty well as well. They're just saying that there's many ways to use these antibodies in cancer, and I don't want you to overlook the ultimate result of this. This is another thing we saw in the ACS posters. We saw that you can use the antibody to image tumors. And so here's a mouse. You attach the antibody to a zinc probe that lights up under a PET scan. You put the antibody in mouse with the tumor, and you can see that you see a lot of the tumor. Remember with these that there's a lot of the tumor in the kidneys. You always have to look past the kidneys to look at where the little arrows are. But the little arrows are showing you that it is already working as an imaging reagent for this pancreatic cancer model. So, how did they screen the surfaceome? This is where you have a complex biochemical process. But I want to show you that you can actually reduce this down to something you can understand. This figure has 4 parts, and the parts are symmetric. So let's start with the first part all the way on the left. You have cells that have just an empty vector. The vector does not have anything in it, and you grow them up. Then you have cells that have the mutated RAS version. And so those cells are turned on. So you have the difference between cells that are not turned on in the red, cells that are turned on in the blue. And what you do is you collect all those cells, you take the protein, you do mass spec of all the proteins. The difference is that you are actually growing up one batch of cells with light isotopes of carbon and nitrogen, and the other batch of cells, the mutated cells, have heavy isotopes. Mass spec can tell whether you have the heavy nitrogen or the light nitrogen in your protein. And so then you get a bunch of peaks. The peaks are exactly the same, but because they're different in isotopes, they're exactly different by the number of extra neutrons that that nitrogen has. Doesn't affect anything else, but it affects the mass spec. This is a brilliant way to be able to directly compare two cell conditions and to see where the proteins are, where you have a lot more of that protein in the cancer cell. And from mass spec, if you remember, mass spec can also sequence the protein. So you take the mass spec fragments, you figure out which protein it was, and immediately you have identified the precise protein that you have a lot more of on the cancer cell. They have another branch to this, but it's symmetric to this. You also grow up in light and heavy, but both of these have the RAS mutant. The difference between these now is you have a drug applied to one of them. So instead of being the difference between normal cells and cancer cells, you have the difference between cancer cells and drugged cancer cells. You have a good drug treatment that goes directly after RAS, and so it turns that down. So this is like a nice little control, because there's so much dynamic, there's so much messiness here. This really helps you have a switch to turn off the RAS and to make the cell normal again. And then you can compare not just the cell with the mutated RAS, but the cell with the mutated RAS turned off. If you get that, you get this figure. Okay? Then this is the results of the figure. And this is called a volcano plot. What you need to look for on plots like that, where you have scatter, you're looking for outliers. Because the outliers are going to be proteins that are either you see a lot of them in one situation, or you see less of, you see them differences in their enrichment. The important thing about this is on the left you have the comparison of the normal to cancer. On the right you have cancer without drug to cancer with drug. And so what you're looking for is you're looking for a protein that's upregulated on the left, but it's downregulated on the right. Or possibly, like I said, you can possibly get something that's downregulated in the case of cancer, but then upregulated. You could use that for certain therapies as well. So look back and forth at these different things, and you see that you have CDCP1. It shows up as a blue protein that's upregulated on the left. It's upregulated in cancer. It's downregulated on the right. When we look up and down, you see it's not just CDCP1. There's also that ITGA3. And then you have some that follow the opposite pattern. They are downregulated in normal, and then they are upregulated in cancer cells. They're upregulated when you apply the drug. It's all about when you apply the drug. I'm saying that backwards. Look at the graph, don't listen to me, okay? But the main thing about that is you can look at those as drug candidates as well if you think through what they're doing. But we know that at the end of the day, the CDCP1 is going to be our answer. So we look for that, and then we understand everything else in light of that. When they look up and down, you can see that they only had a few targets that really followed the exact pattern that they wanted. And they did this for the inhibitor that they have. That's called the MEKI inhibitor, was the one that they used and the one that they showed there. If you notice, there's about 24 dots that show differences in expression in the right direction. And so those 24 dots, great. We've got 24 candidates, right? And so what they did to be really thorough, they know that these are messy experiments and it's really hard to find something that is on the surface and affected by cancer. And so what they did 4 different kinds of experiments. They used 3 different inhibitors, because each different inhibitor is going to produce a slightly different array of stuff on the surface. And then they also used a different, they activated the KRAS, and I believe they used CRISPR for that. They did another method. And all of these methods should have altered the surface based on it being cancer on the inside, making a different surface on the outside. So what you have is that you have the KRAS activation, you have all these different ones, and you only have a few targets that actually show up with multiple inhibitors and with the KRAS activation. It's the blue area right here. And you see you have actually only one that's really in the middle of all of them. You have none that actually show up in all 4 ways of doing it. You have 1 that shows up in 3 ways of doing it, 2 that shows up in another 3 ways of doing it, and 10 that show up both for the MEK inhibitor and the KRAS activation. So they decided to look at those 13. So we've gone down from, we've done a lot of experiments, but we've got 13 targets. And what they did is I believe they made antibodies to all those 13 targets using phage display. One little point right here, this is just a little side point, but I think it's really interesting. A lot of times people tell what's on the surface of a cell by looking at the mRNA, because the mRNA leads to expression, and there is some correlation between the mRNA inside a cell and the proteins expressed on the surface of the cell. Also, you can read out the mRNA, and you can tell which protein has how many transcripts associated with it. Really good stuff. But they wanted to say, why didn't we just use mRNA? Why did we go through the trouble of proteomics? Well, because they thought it would be more accurate. What you're really interested is in the proteins. So you should do proteomics rather than genomics. You see what I'm saying? And they looked at the mRNA, and they found that there was a little bit of correlation, but there were a lot of places where there was not strict correlation. This shows all the proteins that they looked at, all the proteins, the genes that they identified. And the SILAC is what they found with proteomics. RNA-seq is what they found with RNA sequencing. Easier to do, but they don't all go together. The ones that went down in the SILAC assay are red, and you see that the ones that went up are blue. And it tends to, when you line it up with the RNA, you tend to get red with red and blue with blue, especially at the extremes. But in the middle, you get a lot of where it doesn't line up, where the RNA goes down, but the surface expression goes up. And this is just because RNA is not tied tightly. Expression does not mean, or transcription is not translation. You know, it's that kind of thing. RNA is not protein. It's just a proxy for protein. And so they use this to show that there's not much correlation. There's modest correlation. There's value to mRNA studies, but they believe there's more value to proteomics, because proteomics is proteins, and proteins are what the antibody binds to, not mRNA. And so then you have this figure, and this is where they actually do generation and validation of antibodies to, this is how they made the antibodies to their 13 drug targets, or whatever the number is that they actually did. And you see that they actually do a phage display. I want to show you this because it looks like the phage display that we talked about with Delano and Wells. Scientists don't have that many tricks. Once something works, they keep doing the same thing because you want to try to find out where else it works. This is the example with James Wells. He's doing the same thing that he did with Delano and Wells. It's just instead of doing it with the Fc domain, he's doing it with the variable region of the antibody in a sense. And so you have all of this, you have the panning, you have the same kind of things. Don't worry too much about the details, but identify that this is the same method that Wells used before. By the way, when I heard him talk about this, he mentioned something that didn't get into the paper. They were using a FAB library, which is just the variable region of the antibody, the fragment of the FAB region. That's the binding part of the antibody. And they found in their previous work that there was one particular antibody fragment that just expressed really well in their system. They don't know why, but they just keep using it because it works. There's a lot of science we don't know why, but we use it because it works. Okay, so we got to be kind of pragmatic about some of these things. Notice also that the Fc domain is part of this. There's a whole complicated thing about how do they do the phage display. They bind it, then they cleave it, then they wash it, and stuff like that. That's what the Antibot is doing. Because probably it's way too dull even for them to give to a graduate student. All right. So the Antibot is described on this page right here if you want to know exactly how it works. I just want to point out again, you look at these figures, you see phage display. What you learned from that first paper is applied to cancer immunology. Pretty cool. And the one thing they don't talk about at all in the paper, where are they varying the antibody? Well, they're making up they're using phage display to basically make random amino acids, and this shows you where they're doing it. They're doing it at the loops. We said the loops are what contacts antigen, right? So they're mutating the loops, they're doing random amount of loops, and they talk about, there's so many things, there's so much variation potential. They don't even cover all of the possible variants, but they cover a good amount of them. And they try to make a diverse library so that they can have more chances of one that works really well. And then they can pick the one that works best with the tightest affinity. So these papers are also mentioning ELISA. It's one of the things I believe that the Antibot is doing. It's actually injecting onto ELISA 96 well plates. I haven't had the chance to let you do ELISA, but you probably will see it in one of these papers, so I have to mention what it is. Notice that there's ELISA is mentioned on this. They did phage ELISA, and then they took the phages and they DNA sequenced them to see which ones were most abundant. And then they would figure out from that which of the randomized antibodies was working best against their antigen, which was one of the surface proteins that they gave to it. I just noticed something funny down here. They're using protein A to purify the antibodies because protein A binds to the FC region. When you see protein A, you might think, oh, I've seen that before, and they were studying how it bound to the FC region long ago. You never know how something's going to be useful in science. So I just want to say, that's why we're doing these big complicated stories. You never know what's going to come up later from it. So notice right there, after each selection, they looked at 95 different phage clones, and they basically sequenced each of them. Then they ultimately identified 116 unique sequences against 7 different targets. So we're already down to 7 targets because those are the ones that worked well in their system. Bioengineering does not always work for every protein. You've got to try different proteins until you find one that works. That's what I had to do when I was starting off any of my projects. If stuff doesn't work, you don't know why. But if stuff does work, you're happy and you move on. Okay? So they're down to 7 antigens. For each antigen, they have about 100 antibody sequences. And that's because they've been able to do ELISAs. What all ELISA is you put an antigen on a surface, and you turn that well with the antigen in it, colors. You detect antibody binding by seeing if it changes the color of the well. And this also is a primary and secondary antibody thing. You coat the surface with your sample, you add your primary antibody, which is unlabeled, because that's the specific one, and it's hard to label those. You put your primary antibody down, it's got an FC region sticking up, and so you add an antibody against the FC region, the secondary antibody. That antibody you buy in bulk, and you label it in bulk, and you have some kind of label on it. Sometimes the label can just be radioactive, and you scan and look for radioactivity. But who wants to deal with that, right? Much better, actually, what works better is to actually, on the secondary antibody, you have an enzyme. An enzyme that quickly does a reaction. If you have a little bit of uncolored substrate, it turns it into a different color. And you look for the color. So you've got this stack. You've got the sample. You've got the antigen, primary antibody, secondary antibody. Enzyme attached to the secondary antibody makes color. Boom. Once you go through all the steps, you end up with something that, well, here's another way of looking at it, but I'm going to skip ahead to what it looks like. Each of those wells that has colors has antigen in it. You have antibody binding to the antigen. And so that's really cool. You can make a well light up and you can figure out which antibodies have strong binding if they have a lot of strong color. The other thing you can do is you can actually apply one of these primary secondary antibody combinations to a gel. A lot of antibodies will bind peptide regions, by the way, which will persist even when the protein is unfolded. Or you can even run gels on folded protein. You just leave out the SDS. So you have a case where you blot the gel with the antibody. And you make that specific antibody binding site light up. So here you have a case where you have an SDS gel. You have the immunoblot on the other side. The SDS gel shows that there's a lot of proteins. Like, for example, lane 1 is a lysate. But you take and you blot it. In lane 1, you see that the antibody is only binding to that one protein. It's just not that concentrated. But as you went to lane 2 and lane 3, you purified the protein more, and you ended up with purer protein. And you've shown not only is it the right size, but you show that it is definitely the right protein because you have a specific antibody binding event there. So you can do this with a micro well plate with 96 things. You can do it with a SDS Phage gel with 10 lanes in it, or you can do it on a large scale with what's called an oh boy, they don't have the name of this, a microarray. It's basically like a 4,500 well plate, and it works by the same idea though. And what you have is you have each of the dots lights up with the labeled secondary antibody. It lights up more if you have more antibody binding. Here are sera from a US adult that has never been affected by malaria on the left, and there is a malaria infected individual serum from West Africa on the right. Basically, they took 4,500 proteins from the malaria parasite, they put them on the chip, and they exposed it to the serum. So this is polyclonal, actually. And then they applied the secondary antibody. You see why the secondary antibody is so helpful here, because all of those polyclonal antibodies have Fc regions that will bind the secondary antibody. It comes down to a simple readout. The serum from the malaria patient lights up because it has a lot of malaria. It reacts to a lot of different malaria proteins. Which one does it react to most? Maybe we want to use that protein in therapy, or maybe use an antibody to that protein as serological therapy. So, we might do a CRISPR Journal Club later. We probably won't have time, if I have to be honest, but there's a whole section from the 2017 Wells paper where they talk about a CRISPR Journal Club. They talk about how CRISPR was used. And that was that other way of looking at things. So they used a CRISPR assay, and it found, they basically knocked down different membrane proteins, and they tried to see if they detected that going away or not. All that is to say, and this was connected to oncogenic grass. This paper is already big enough, but the thing I want you to see is they used another technique, a CRISPR-based technique, to really go after, and they tried to find something that showed up in all their techniques. They did find something. They found two somethings. Both of the things they found were great. But you think about how many thousands of proteins were ultimately scanned. I wonder if they were a little disappointed, honestly. But at least those are two good possible targets. In fact, one of them, ITGB5, I believe it has already been implicated, and they had reason to believe other people were already working on it. But the CDCP1, it looks like there wasn't as much work going on it, and so after all that funnel, he talked about the funnel of drug discovery, right? There's a similar funnel for antibody cancer drug discovery. But they ended up with CDCP1. They looked in different places to see, is this really associated with cancer in other people's experiments? And they found lots of evidence that it was. And they were able to find sort of the needle in the haystack. It looks like a really good target for particular kinds of cancer. And that is the end of part 3. That's how you can use antibodies against cancer. Next up, we're going to focus specifically on SARS. But I've talked enough today. Let's send this off to YouTube.